next speaker is Ben Ball. The title of his talk is Circadian Clock Control Gene Expression in the Ovary. Implications for the timing of ovulation.
Good afternoon, everyone. Like Professor Hirsch said, my name is Benjamin Ball, and I'll be talking about the work I've done for the past 8 months in the Menneker laboratory, researching clock-controlled genes and their role in the timing of ovulation. So first, what exactly is a circadian rhythm? Well, circadian rhythms are endogenous cycles of physiology and/or behavior and gene expression that have periods of approximately 24 hours. Now, in mammals, the suprachiasmatic nucleus, or SCN, is what's known as the primary pacemaker, or the central clock, and it controls these circadian rhythms through receiving environmental information, cues about the light-dark cycle, and entraining these rhythms to the environment. Interestingly enough, these rhythms have been identified in other tissues throughout the body, and these tissues are known as peripheral clocks. The SCN also has a role in training these clocks and synchronizing them to each other. At a cellular level, this mechanism is brought about by an autoregulatory negative transcription translation feedback loop. Large words, but really what we're talking about is that the transcription factors clock and BMAL are rhythmically transcribed and they heterodimerize. This complex goes back into the nucleus where it binds to EBOX elements in the promoter regions of genes, and it upregulates transcription of those genes. Period and cryptochrome, two such genes, are therefore rhythmically transcribed. In the cytoplasm, they are phosphorylated by casein kinases. This complex heterodimerizes and translocates back into the nucleus where it decreases the activity of the clock BMAL heterodimer, thus completing the negative feedback loop. This clock is responsible for controlling a lot of physiological rhythms, including body temperature, kidney function, cardiovascular function, and on the right you can see examples of endocrine function. One of the best studied examples of endocrine function is actually the timing of luteinizing hormone in the cycle of ovulation. So in the rodent estrus cycle, or ovulation cycle, we have a 4-day period, 2 days of relative quiescence, metaestrus and diestrus, through which time these black dots represent increasing levels of estradiol. The levels of estradiol peak on the day of proestrus, which causes a surge in luteinizing hormone, these white circles here. This luteinizing hormone is going to travel to the ovary, where it induces follicular rupture, also known as ovulation. This process takes 16 to 18 hours, and therefore estrus, the last day, is the day of ovulation. Classically, this whole process is thought to be controlled by the circadian rhythm, which passes on its information about the light-dark cycles to GnRH neurons, or gonadotrophin-releasing hormone neurons. These neurons cause the pituitary to secrete luteinizing hormone, which circulates throughout the blood, travels to the ovary, and there we've seen that it causes ovulation. Now, in this model, it's important to note that the SCN has been implicated as driving the entire thing, and the ovary is simply a passive responder to this model. It's interesting to note that the new work in the Menneker laboratory has shown this might not be the entire case. There are other facts this model can't explain. So I'm going to take you through this experiment here using luminescent reporter strains of rats. We have here ovarian cultures of these rats, and this shows a rhythm, a circadian rhythm of per1 expression over time. Now, this rhythm damps, but it can be reestablished with just a simple media change. Here we have a picture of the trough of these luminescent cells at the trough of their intensity, contrasted with them at the peak of their intensity. And each of these plots is an individual circadian rhythm of per1 expression, pixel intensity, from individual cells. So taken together, this data really goes to show that there's a circadian clock in the ovary. Now, to test the role of the circadian clock in the ovary in ovarian physiology and the timing of ovulation specifically, the Menneker laboratory conducted another novel experiment. In this experiment, they blocked endogenous secretions of LH with a GnRH receptor antagonist, and then measured the ovulatory response to timed injections of luteinizing hormone. So in the graph here, we have the mean oocytes produced per animal as a function of the time of day. And the lighter portions of the graph refer to the light phase of the light-dark cycle that the rats were kept on. The dark phase refers to the dark phase of that light-dark cycle. So as you can see here, the data shows an actual diurnal rhythm of ovulation in response with a larger response seen on the dark phase of both diestrus and proestrus. So the actual peak occurs, interestingly enough, on the afternoon or evening of proestrus, and that's the normal time at which the LH serve is received physiologically. So this data has been repeated in rats kept in constant dim light and in mice kept in constant light-dark cycles, and the similar results are seen. What this indicates or suggests is that the ovarian clock may play a role in setting a window of sensitivity to that so that the luteinizing hormone, when it's received, it only has a great function if it's received at the appropriate time for the circadian clock. Oop. Slides. Okay, so this mechanism is still not understood as to how the clock could potentially control this. It is known that the circadian molecular clock drives rhythmic expression of other genes. These are known as clock-controlled genes, or CCGs, and they're thought to be cell and tissue specific. So one hypothesis is that the LH receptor, or chorionic gonadotrophin receptor, LHCGR, is one of these clock-controlled genes and is rhythmically transcribed in ovarian cells and causes this modulation you see. So here's the model drawn up. What we've seen up here is the same thing that we talked about earlier. It's clock, BMAL, cryptochrome, and period. Down here we have clock BMAL binding to an E-box element that may be upstream of the LHCGR receptor, causing rhythmic transcription. This 7-transmembrane domain receptor is going to translocate to the membrane of the cell, and there it modulates the response to luteinizing hormone. So just to reiterate, the hypothesis that I was testing was that these clock-controlled genes in the ovary, specifically of luteinizing hormone receptor, are going to play a role in this timing of ovulation, setting this window of sensitivity. So to do this, I took adult cycling period 2 luciferase transgenic transgenic mice, housed under a 12-12 LD cycle, and on the day of proestrus, I euthanized them, and through a needle puncture procedure, I cultured these ovarian cells, shown here. These cultures were synchronized after 4 days of incubation with forskolin, which is a known promoter or inducer of period 1 expression, which has been shown to synchronize clocks. Then, beginning right after forskolin treatment, which was our new ZT0, and then 4 hours afterwards for 24 hours, I extracted the RNA and quantified it with quantitative real-time PCR. So this is some data from a bioluminescence recording of period 2 expression in these ovarian cell cultures. Figures A and B are taken from cell dishes that were cultured at 250,000 cells per dish. C and D are taken from 750,000 cells per dish. And what you can see here is that this forskolin treatment is really creating circadian rhythms of period 2 that last at least 7 days. The periods of these are between 24 and 26 hours, so appropriate circadian rhythms. And because similar rhythms are seen in the 250 cell culture dish and in the 750,000 cell culture dish, it's evident that cell density doesn't have too much of a factor in this. Either cell density is appropriate. This is the data taken from the actual qRT-PCR reaction. So all this data is compared, it's a relative fold change to the data shown here at ZT0. Low amplitude circadian rhythm of LHCGR mRNA expression over the course of 24 hours. It starts off by decreasing to about 2.5-fold of that present at ZT0, and it actually raises up to about 1.5 to 2-fold that seen at ZT0. So there is this nice circadian rhythm of gene expression going on here. So conclusions from this. Well, we've effectively shown that forskolin induces PER2 expression in these cells and is therefore synchronizing the molecular clock. We've also shown that there is this low amplitude circadian rhythm of LHCGR mRNA, and that it's very possible that this rhythm and potentially several others are playing a role in setting this window of sensitivity we've been trying to investigate. I'd like to thank Professor Menaker and Dr. Mike Selix, without whom I never would have been able to do any of this project. So, thanks.
Question for Ben.
Yes.
How is it forscolin's inducing the PER1 expression?
I didn't show the data taken from another paper, but forskolin is an adenylyl cyclase activator, and it increases levels of cyclic AMP, and that's been shown to induce PER1 expression. And once it's ramped up, that component really drives the rest of the clock to synchrony.
I was wondering if you could go back 2 slides. Somebody.
2 slides to the end?
The end.
Oh, you want to give it to the end? Thanks.
The one with the, is there. That
Okay.
way. So if you didn't know ahead of time that it was cyclical like this, would you interpret the data that way?
If we didn't know ahead of time, well,
I mean, you have this expectation of this sort of, it looks to me like level A, level B.
well, we were hoping to see something like this. So if that's what you're saying, we might be interpreting the data. Basically, what's shown here is these are every 4-hour interval. So between interval from ZT12 to ZT16 is the same amount of time from any other interval. So with that in mind, the level really does, like you said, it drops to almost level A, and then it rises up to almost what can be seen as level B. But the important part is that this is over the course of a 24-hour period, and it seems to end up almost back at the level where it started. So that's the important point with.
So it's not that you're assuming that it's being made in these sort of oscillatory amounts.
Over the course of 24 hours, we see that we see an oscillation essentially. The important point to take away here is that the levels are going to be 3-fold less at some point, and then they're going to raise up to about 2-fold more at some point. And it's over 24 hours. So the actual fact that it seems around from ZT4 to 12 to be at one level and from ZT16 to 24 to be at another level, it's really supposed to be taken within the context of this is a 24-hour period total. Does that answer your question?
You totally answered my question. I'm not totally sure I buy your answer.
Okay.
Yeah, I'm fine, thanks.
If I can follow up on that, I think maybe a follow-up question here is, did you repeat the analysis here? Are these single observations, or do you have...
This data is taken from one experiment. We had a little bit of trouble with primer design. Melt curve analysis, we're showing several isoforms of LHCGR were being amplified. We did a primer redesign, and we ran gels, and it seemed like we were getting one product, and then we did the RT-PCR on it, and unfortunately, the data was very questionable. The melt curve analysis was terrible on the second run. So this is only the data from the first experiment that I've conducted.
So each dot is a single measurement. It's not composite of multiple measurements.
Each dot is actually taken from three samples collected from the same relative ZT point.
Same one biological sample tested 3 times by the PCR.
It's 3 biological samples cultured from mice and taken at these points, and it's averaged to get these numbers, and then normalized to an internal standard 18S ribosomal RNA.